chromatography for various applications. I'm going to go over this technique for you guys. This is a fairly informal presentation, like there's no script to go off of. So if you have questions, feel free to interrupt and ask them live. Sharing the presentation with me is Lily. I'll also let her introduce herself next year. Oh, I think you're muted, Lily.
Sorry, I forgot to turn on the mic.
No worries.
Hey, I'm Lily. I'm a third-year PhD student in environmental engineering. We also use HPLC as a analytical instrument all the time. So yeah.
Cool. All right, so to get everyone interested, or like provide some background of why are we even learning this. You might not know it, but HPLC is really a cornerstone of living in the modern world. Most of all pharmaceuticals and drugs that are passed by the FDA have to go through HPLC quality control. It's used as a quality control instrument. It's a series of tests to make sure that the Tylenol you take or the Benadryl that you take is not contaminated with other things. That's one of the big applications here. More related to you Shine students, this is a very highly desired skill set for undergrad research labs, especially for chemistry, biology, and environmental engineering, if you're interested in those. Finally, longer down the road, you can actually make this one of your professions if you want. There's highly lucrative engineering positions for liquid chromatography specialists. Here's one I found in San Francisco. That's the pay range. Not too bad. So we can move on to some of the basics that you need to understand. So one of the most basic properties for liquids or solvents that you work with is the polarity, or meaning if the solvent is either aqueous or organic. So the classification depends on if the molecules dispersed in your solvent have differing charges, meaning they're polar, or the opposite, where they don't, if everything is kind of balanced. So over here on the polar side, we have chemicals like hydrochloric acid, which the chlorine here will want to attract electrons more than this hydrogen, so it's more negative. Obviously, water is polar, and some of the other non-polar organic molecules are on this side. So if you mix a polar and non-polar solvent together in sort of a funnel, this is called a separation funnel. If you shake it up and let it sit for a while, they'll actually separate into two layers because of this polarity difference. While density does have some part to do with it, it's not only based on density. So if you have a layer of something less dense than water, than the aqueous condition, it'll rise to the top. But you can also have the opposite, where some organic solvents are heavier than water and will sink to the bottom layer. And knowing that, we can move on to describe what chromatography is itself. The word itself means sort of color science or color descriptive science. Many of you may have done the simple kind of elementary school science experiment where you dot a tissue paper with some ink and then immerse it in water. And what you'll see happen is the water front will move past that drop of ink and pull different portions of the ink apart from each other. The portions that are highly bound to the paper will remain kind of lagging behind, while the more soluble things will be pushed along, and you get this kind of nice smear of different components. This is also classically done with mushed up plant parts. You can separate the chlorophyll components in the tissue paper as well. This is known as paper chromatography. But in our lab, we use a more commercialized version of this called the HPLC, or high performance liquid chromatography. This is a picture of what the whole setup looks like. Hopefully by the end of this lecture, you'll see more than just a series of boxes. But we have several uses for this type of machine. We can purify or isolate some particular chemical in a sample, analyze the properties of that, like the concentration you have it in the sample, molecular weight, other properties like that. And finally, as I said earlier, quality control of different pharmaceuticals. So what these series of boxes are is actually a flow loop of liquid, which is simplified in this diagram here. What you have are basically starting off with some solvents. We have one for organic and one for aqueous, if you recall from that previous slide. These are mixed together in appropriate ratios and pumped through where you inject your sample here. And this is finally passed through a column, which is the part doing the chromatography. And finally to some detectors that'll look at the color of your sample. It's more robust than just the color. So you're actually going to detect particular wavelengths of light going through your sample, but it's basically the color of your sample. And our boxes that you saw earlier are kind of self-contained portions of this flow diagram. But the first one was the solvent reservoirs and the mixing. It's one box. We have two pumps, one for the organic and one for the aqueous. The column itself is in a temperature-controlled oven in its own enclosure. The detector as well as the final output. So here you can either put the samples to waste if you're just analyzing, or you can also collect portions that you want for later studies or later purification. This is just overlaying that flow loop on our actual setup. So the solvent bottles are up here. Two pumps down here goes to our injector, which mixes our sample with the aqueous or organic solvent. The column is kind of like a thin tube of metal in here. I'll show you guys later. Back up to the detector here and out to the waste here. Yeah, any questions so far? I don't know. Looking back. All good, so we'll move on to this. So yeah, going to the actual meat of how this separation is done. That column box you saw earlier contains one of these round tubes. And this is where your organic and aqueous solvent is passing through. Inside this tube is a packed sort of sand or silica that is the actual filter material. So on the right here, you have some scanning electron microscopy images of that material inside. You can see there's pores in between this silica bead that is allowing the aqueous portion to flow through. And the silica itself actually has a roughness which will depend on the manufacturer as well as what application you want. If you want it to be high binding or different chemical reactivities. This here is a slice of actually cutting the column in half, and you can see the bead in there packed tightly. A lot of research R&D is spent on optimizing this formulation, and companies can do very well or very poorly depending on how they do this process. So this column is generally referred to as the stationary phase because it doesn't move compared to the mobile phase or the liquids that are passing through. Yeah, so here's just a diagram schematic of what would happen if you put some like generic black ink into a column with flow running. You would separate out the different colors of that black ink. So supposedly your blue part of your ink is interacting with these silica beads more, and they'll flow through your column more slowly. And this is kind of like what happens in that paper chromatography we said earlier. And this is representative if, say, the chemical you want is sort of one of these bands only, and the other colors are you want to reject or to purify your sample. One of the most common types of HPLC that is used is called reverse phase HPLC, which means the column itself is hydrophobic or nonpolar. So if your sample is nonpolar, it will stick to this column more than the polar portions. The mobile phase instead is polar and water usually. This method is used more commonly since the mobile phase is not as toxic or messy to handle as if you had imagine it's like an oil or like an organic flow loop. Most things in biology dissolve in water, so we can use this type of HPLC. The normal phase is just the opposite. I think it's only named this way because normal phase was discovered first, but for some reason, reverse phase is much more common. And the normal phase HPLC, your column beads are polar, and anything non-polar will flow through faster. So there's that. And moving on to the next box, we have our UV-Vis detector, or basically the light detector, looking at the optical properties of the fluid going through. So we don't just look at the color in the visible spectrum for our machines. We also look at some shorter wavelength light, like as low as 100 nanometers. This is still the same thing as color. Basically, just because your eyes can't detect this electromagnetic wave, your samples will still have very distinct properties in this region. So what's known as a UV, visible detection, by this light detector. Yeah, our machine uses a prism to basically isolate some regions of light to shine onto your sample and see how much passes through. And basically that's the darkness or opaqueness of that sample. Yeah, so some of the different commercially available machines are for different scale operations. What you'll hear most commonly is analytical HPLC, which is when you're injecting samples that are very small, sort of maybe like just for analysis. If you're taking some blood samples and you want to see concentration of a drug inside the blood, you're not going to be taking like liters of blood from a human. You can only get very small samples. So you use an analytical type HPLC to look at that scale. The other end is preparative HPLC, where if you made like 10 gallons of this chemical that you want to purify, you can't be putting like 10 microliters at a time through the analytical. So you put it through a much bigger system which a high flow rate, high pressures, much more optimized for purification. These generally have less resolution, but you're getting much more product. And kind of in between, the jack of all trades, but master of none, which is the system that we have, can sort of do both, but it's not like super accurate as well as not super large scale. So semi-preparative is the name for that. Yeah, finally, you can see the overall flow of organic solvent over time in this graph. This is what's known as a chromatogram as well. So the blue graph here, first of all, the x-axis is just time, or in this case, it's the column CV or column volume. If you have a constant flow rate, this is just increasing over time. So you can treat that as the x-axis. The y-axis here is going to be the percent of organic solvent you're putting through. So as we flow the HPLC, we actually increase the amount of organic solvent, in this case, acetonitrile, compared to water over time. And this will actually draw out different parts of your sample coming off the column at different rates. The blue curve is not part of this axis, but that's just if you were looking at a specific color of your sample, you get different spikes corresponding to different chemical products coming out at different times. So the ones later off have higher affinity to your column. So those only come off when your acetonitrile is much higher, like 80% of the ratio of water. And we usually bring it back down to equilibrate back to like mostly water as well. And here's a interesting timeline of just the whole HPLC field. Usually the timelines are given at the start of the talk, but I think it's better if you know some background before. So chromatography as a field it was started off in the 1900s by a Russian botanist named Mikhail Svet. And he was looking at different plant pigments and separating them in paper chromatography. It wasn't until the 1960s until the first commercial HPLC was made by the Waters Corporation. Actually, these blue highlights are companies that you might want to Google later on if you jot down their names. They're very big names for HPLC and chemistry in general. They definitely offer internships for high school students and college kids. Maybe not now, but in the future, they will offer positions for HPLC or analytical chemistry. So Waters was the first company to make a commercial HPLC. Next in the 70s, Agilent Technologies introduced a very good light detector to look at the chromatography wavelength properties of samples. And that was a much more rapid method of chemical analysis. In the 80s, there were much more kind of stepwise improvements to the whole system, increasing pressure and resolution of your samples. And the 90s, the decade I was born, if you can imagine, Waters introduced the Alliance HPLC system. So that was mainly targeted for pharmaceutical sciences. And it looks very much like the picture that I showed you in our lab. And it became the gold standard for HPLC. And it's named as one of the most successful products in the history of analytical instruments. So that's not a light thing to be able to earn. In the 2000s, Agilent, 2006 specifically, Agilent introduced the chromatography column to remove the most abundant proteins in human plasma. So for the first time, you could see very small, previously undetectable proteins in human studies. So that was a landmark technology. 2008, a company made some improved fittings and tubing setups for increasing the pressure and ability to separate different components as well, called IDEX. Finally, 2010, a company called Phenomenex. This one is much smaller compared to the other outlined ones, but they're actually local to Torrance, California. And they made sort of the world-class columns that we use called kinetics, core shell columns. So we actually buy the HPLC system from a different company, discard their columns, and buy Phenomenex columns. So that's kind of how the state of things are now. So they offer the best separation technology. So yeah, one of you might work for these companies later on in your career. So yeah, with that, I'll hand it over to Lily, which will go over specific applications of this in lab. I'll stop sharing.
Mm-hmm.
Yeah. Any questions so far? If we take a pause here.
Okay, so I'm gonna take over and talk about the specific applications of HPLCs. There are many different types of applications of HPLC in different fields. All in all, they're widely used for identification and quantification of organic compounds and different metrics. For example, in pharmaceutical world, people use it to measure antibiotic, sedative, steroids. And in biochemical engineering world, they usually use it to measure big biomolecules like amino acid and proteins and carbohydrates and lipids. And food products, people use it to identify artificial sweeteners and antioxidant, stuff like that. Actually, there's a huge HPLC lab in Pepsi and in Coca-Cola. They use it to find new flavor compounds. And for monitoring pollutants, like EPA, Environmental Protection Agency, they use HPLC to measure pesticide, herbicide, and PCBs all the time. And for cynic chemistry, people use it for toxicology usage, like they measure drugs and poisons in people's blood sample, and that can use to measure blood alcohol levels as well. So in my area of research, which is environmental engineering, we use HPLC to look at micropollutants. So micropollutants can be defined as the anthropogenic chemicals that occurs in aquatic environment well above a natural background level due to human activity, but the concentration still remain at a trace level, which is very small. For example, micropollutants include disinfection byproducts, personal care products, pesticide, pharmaceutical compounds in wastewater and in drinking water. Disinfection byproduct is formed by adding, for example, chlorine as disinfectant, and it will react with the organic matter that is primarily present in the water body, and it will form disinfection byproducts. And compounds like personal care products and pharmaceutical usually are discharged through human excretion. Like people use those products or eat those products, and it will end up in our freshwater system. So in order to quantifying those compounds, because we're interested in how big of a level, that means concentration of those compounds are present in the water sample, there's a common practice that is called a standard curve. So we usually make standards with knowing concentration, but at different levels. So you will make 5 to 6 different samples with different concentrations, and we run them through HPLC. And then the response we got for each sample is called a peak area. So we plot the peak area against the concentration, and we should get a line across the original point, like shown in this graph. The line should be, it should be a perfectly linear line because the peak area should be in linear relationships with the concentration. And then we can run the sample with unknown concentration, and we'll get the peak area of that sample, and then we can line that peak area to the curve that we got, and we can know the concentration of the sample we're measuring. Does that make sense? Cool. Also, people use it to identify new compounds. In this way, it's more of an identification purpose instead of quantification. So that should be something you don't know what it is prior to your analyzing. You're just looking at new peaks, basically. So it depends on what kind of detector you're using. For new compound identification in our lab, we usually use a mass spec. So it's not a UV-Vis detector, it's a mass spectrometry, which measures the weight of a compound. So if you're seeing a new distinct peak with a new mass, usually that means you are seeing a new compound. So you're identifying new compounds, but that can also be done through UV-Vis. So usually we look at the 3D plot of the result, which is shown in the colorful graph on the right. So you can see on the x-axis is time. So if you are seeing like multiple peaks, usually that means it's different stuff. So because every compound has their own retention time based on their polarity. So after you see a new peak, usually we go to the other side to see the UV spectrum of the compound. So for each compound, based on their chemical property, will have a different absorption peak. So for some compound, the distinct peak will be located at certain wavelengths. So for example, this one, the one we're looking at, has 4 different absorption peak at 260, 350, 370, and 400. That's very special, and it's probably only for this compound the spectrum is going to look like this. So that's what we usually do for new compound identification purposes, just to look at the DAD result of the compounds of the new peak you're seeing in your results. We can also use it for purification, because there is another add-on component of HPLC that is called fraction collector. So it's something added in the end of the HPLC after the sample are running through the column. If you don't have the fraction collector, everything will just go into the waste with the eluent with your mobile phase. But if you have this fraction collector, it can collect the part that you want. For example, we're now looking at 4 different peaks. That means in the sample, it has 4 different compounds, but it's a mixture. But you only want the second one, the one at the 1.7 minute. So you will turn on the fraction collector and tells it, so I only want you to collect the thing that's coming out of the column from 1 to 2 minutes. And it will, like, the rest of the sample will go into the waste, but the part you ask the fraction collector to collect will be stored in a small vial. So theoretically, the thing you collect will only contain the analyte you're interested in for purification and some of the mobile phase. So the mobile phase will be organic solvent and water, and you can do further like evaporation and stuff to extract the sample you want. So that will be what we do if we want to separate one particular compound from a mixture. So now I'm showing you some of my real life data. So for my project, the way I use HPLC coupled with a mass spec is to measure drugs in wastewater, because a lot of people are taking illicit drugs such as methamphetamine and like ephedrine, stuff like that. Those compounds cannot be fully absorbed by human body. So at certain level, it will be excreted through urine and end up in our wastewater. So I collect wastewater samples from wastewater treatment plants, and I use LCMS. The LC part is the liquid chromatography, and the MS is the mass spectrometry, to quantify how high the level of illicit drugs that are present in our current wastewater. We use mass spectrometry because usually HPLC with UV-Vis, it doesn't have that high level of detection limit. So mass spec is more sensitive for very small concentration measurement. And you can here look at, that's the real life data I got. And you can see the peak we're seeing is for methamphetamine. And it increases with respect to concentration. So the one in the bottom has like a higher level of meth that are present in the water. So you usually see something called a TIC, that stands for total ion chromatography. That is that's all the signal that the detector is receiving from your samples. And you extract the mass you're interested into an EIC. That's what we're looking at. So I asked it to only look for the compound that is weigh 148, which is the mass of methamphetamine, and it will extract a peak for you. And you will use a reference standard to confirm the retention time. So if we purchase methamphetamine and make it into a standard, the peak should also show up at 1.4 minutes. So you use the retention time to confirm, also with the mass, to confirm that is the compound you're looking for. And then we use the peak area you are getting here to determine the concentration level. So, but we have, here I'm gonna talk about some of the common issue that we're usually facing when we're using the instrument. So for HPLC, because it's a liquid chromatography, we usually are expecting a little bit of a matrix effect. That means if you're using it for real life samples that is in a dirty matrix, such as wastewater or like human like blood, you're gonna see a little bit change in retention time and response compared to your analytical standard that is just dissolved in DI water, because there are other compounds that are present in your real samples, like salts and stuff. Sometimes that will change the response in a certain way. So occasionally it's not very stable, but most of the time it's fine. So if you're expecting a strong matrix effect, for me, especially when it comes to wastewater sample, because it's pretty dirty, what we usually do is instead of using a standard curve that are made in DI water, we'll remake the standard in a addictive way in wastewater matrix. So you would just spike in the compound into a wastewater sample and then remeasure the peak area and use that as your new standard curve instead of just using the old one you made in DI water. And then the second common issue we usually come across is when you're trying to set up the method, you need to adjust the mobile phase ratio. So there are two types of mobile phase. One is the organic phase, the other one is the water phase. So it depends on how polar your compound is. You need to kind of adjust the ratio, because if your molecule is too polar, usually it doesn't retain very well, so the retention time will be very short. So sometimes you will get like crappy results. But if your compound is really non-polar, as mean it's really sticky, sometimes it will stuck in the column and it has a really long retention time. Like if you're using the same method as a rather polar molecule, the retention time will be like 3 hours, and that method will be insanely long, and that's not reasonable. So you need to adjust the ratio. Usually when we do if the compound's like bigger, like some of the long chain hydrocarbons, we will increase the ratio of the organic phase, like acetonitrile or methanol. So you usually need to adjust the ratio to get to the retention time you want, which means you don't want it to be too short. That is just like it's not going to retain a column at all. It's just going to be shoot right out. That's not ideal. You also don't want it to be too long. That means your method is going to be very long and it's not very time efficient. And then the next common issue is just the compound is too polar because we use reverse phase HPLC. That means the compound is not going to retain at all. It will just be too wellly dissolved in water and will just shoot right out of the column. In that way, you probably need to change to a new column that has a different filament in it, or find a different way of measuring it. So that will be kind of a nightmare if the compound is not retaining at all. Usually that means the compound is probably not very good to do HPLC analyze. And the other one is there are many different compounds that is in your sample, but it cannot be separated very well. If the compounds have too similar of a hydrophobicity, so this will be a ideal situation. There are 1, 2, 3, 4 different peaks. They're all very well separated. The peak is very sharp. That's what we usually want to look at. We want the peak to be as sharp as possible and well separated. But sometimes it will look like this. So that will be 2 peak merging into one. That's usually going to give you a hard time on getting a very precise result. So if the compounds are not, the separation is not very good, you're getting like a merged peak looking like this. You can probably get away with it by changing your mobile phase ratio and flow rate, but sometimes you just don't have any luck and the two compounds are just chemically too close to each other and the separation just can be done. We will split the peak like the one we're seeing right now. So you would just, instead of doing integration on the whole peak, you would just find a middle place that will just cut the 2 peak apart, and you would just get the area based on this. But this is not an ideal situation. And the last biggest problem is just maintenance. Usually an HPLC is kind of expensive and it's very delicate and it needs a lot of work to be taken care of. So it's like a plumbing system because it's a lube. They're like fluid moving around in it. So we're very afraid of leak and blockage. So in leak, that means just water droplets is gonna sipping out from somewhere if you're tubing, and that's gonna damage the device. That's something you don't wanna see. You also do not want any blockage. That means if you were introducing particle into your system, the column looks like it's kind of big, but there's a little particle that is inside. The perimeter is actually very, very small, so you can't have anything that is solid that will be introduced into your system that's going to block the whole column and damage the system. You probably, if there's stuff stuck in your column, you need to get a new one, which is kind of sad. And it's also need to be airtight. So sometimes it happened in our lab several times. Some people would forgot to turn it off, and it will keep pumping overnight, and it will use up all the mobile phase that is in a bottle. So and then air was starting to be sucked into the pump and into the column, and it will somehow damage the system too, because the pressure system will be off. Sometimes if there's air bubble stuck in the pump head, the pump won't work properly. Or even worse, it will stuck in the column, and then you will have a pressure wobble all the time. And then your compound and your peak is gonna start to look really ugly. It's gonna be wider and wider, and sometimes the peak will be splitting for no reason because the air bubble is causing that split. And usually that will be a pain. You need to sonicate the column and trying to backflash it, and sometimes just will ruin the column and you just need to get a new one. So yeah, that's usually the maintenance issue that we come across during using it. The component, like the component of HPLC usually are very steady, like they don't break very much, but definitely the plumbing is the thing we usually do when we're trying to do maintenance on the system. You just always need to make your loop very clean and everything can go through and there's nothing stuck in there, stuff like that. Yeah, that's pretty much all I want to cover on this. So any questions? Good.
Yep. All right, I guess we'll wrap it up. Thanks everyone for attending. Yeah, I think this is recorded and you'll see this on the drive if you want to go over anything. Feel free to reach out to either Lily or I for email questions or anything that comes up about HPLC. Cool. All righty. Thanks, everyone.